Yeah, I'm very happy to be here. Thank you so much for organizing this. So I start immediately because the time is strict, I've heard. And what we know from the European waters is that they are very variable. And this can be both when we look like comparing north to south, but also when we're comparing winter to summer. So we really have certain change in temperature, for example, but also when we look at salinity. And salinity is the main thing that is influencing the Baltic Sea. The Baltic Sea is up here, where we are coming from, but also like Nilla's research takes place, and where we have here heard already about Christineberg. And what happens there is that we have a very strong salinity gradient. So when we have here fully marine salinity, then here in the eastern, the northern part of the Baltic, we go down to almost freshwater conditions. And you can imagine this is an enormous stress for animals, and only very few fully marine animals actually manage to fully colonize the Baltic Sea. And therefore, we can also see an enormous drop in species diversity in the Baltic Sea. And one of the animals that made it until this very end almost of the Baltic Sea is the broad-nosed pipefish, Syngnathids tufle, which we have already heard about before. And it has a very broad distribution across the European coasts and also managed to go into the Baltic Sea. And this means that most likely it must have a very high range of phenotypic plasticity, and also that it can locally adapt to environmental conditions. However, we have to admit that the Baltic Sea is only about 10,000 years old, which is a very short evolutionary time for adaptation on a genomic basis. So when we have now changes of abiotic factors such as salinity drops, but also heat waves or temperatures that are different between locations, then it on the one hand influences the animals. This can be in a sex-specific way, but it also influences other biotic factors such as the parasite prevalence and the microbiome. And these then again feed back on the hosts such that we have really an interaction of genotypes of the microbiome, the parasite, the host with the environment, which can have severe implications, which means that we have to look both at these biotic factors and also at the abiotic factors, how they are influencing the host. So this is what we aim to do, and I want to present you two short studies. One focuses on seasonal dynamics in the wild, where we have been sampling pipefish for 4 years over the breeding season every week. Here just in Kiel, so that's Falkenstein, our local population. And then the day has come to have the first Eurosync population genomic results. Isn't that amazing? So keep you posted, but first we go through this one, okay? So this is just when we have the breeding season of the pipefishes from April to August, where you can see the temperature profile that ranges like from 6 degrees up to 20 degrees, and also salinity changes. And when we now went through this sampling every week, then we can compare females, males, and pregnant males. And what we can see when we look at the body condition index is that it really matters on whether they are reproductively active or not. Because in females that are producing eggs throughout the breeding season, as in pregnant males, throughout the season, actually the body condition is decreasing. So they are suffering because parental care and egg production is costly. And this is also reflected in the parasite prevalence that they have. So when we are counting these black spots that you probably all know, these trematodes on the pipefishes, then what we can see is that the non-pregnant males that are not reproductively active, that have a much lower prevalence of these trematode parasites. So if you have the same animals actually do transcriptome-wide gene expression analysis, so we look which kind of genes are actively expressed during the different time points of the season and compare males and females, and then look how these biotic, like parasites, and abiotic factors are potentially driving these effects, then what we can see that on the first PC, which means the part explaining most of the variation, there is no sex influence. But it is really these abiotic factors that are massively driving gene expression patterns. So it is the time, so the seasonality, it is the temperature, and it is the salinity. And when we then look what kind of genes are actually involved here, then we can see that it is the metabolic processes that are highly influenced with this. So our animals, depending on the time and of the season, of the temperature and the salinity, they're changing their metabolism, which also makes sense given that we can see this decrease in body condition index. When we go on a different PC4, so this is less variation explained, but still enough variation, then we can here see our sex effect. So there are expression differences between the males and the females. And those seem to be driven massively by the two parasites that are very prevalent in the system, so by Urodactylus and by Cryptocotyla lympha tematode. And also by the salinity and the body condition index. And interestingly, here the genes that are actually driven to be expressed are those that are involved with the immune system. So it seems to be a very intense connection between sex, the immune defense, and the parasite prevalence. And in addition, which is supporting this, is also the reproductive microbiome, because here we can actually find exactly the same patterns. So to make this short, the reproductive activity is also associated with shifts in the microbiome. So we can see here that the females in red have a different reproductive microbiome, early mid-season to late season, and the males are changing their reproductive microbiome massively during the pregnancy, whilst the non-pregnant males, they remain constant. Altogether, this suggests a likely scenario, we think, that we have a very costly parental care and egg production. This is decreasing the resources that are available to be invested into immune defense. So we do see a trade-off. And this also leads us to reduce parasite resistance and a shift in the reproductive microbiome. And now the time has come, and we change to urosync. So what you have all been involved in, most of you, is the collection of the animals. And then, basically, these very tiny, but I'm very happy to take larger pieces of DNA in the future, of tissue in the future, have come to us, and we do DNA extraction, and then we do sequencing of all these genomes. And I will only show you the very first insights, and only to Syngnathids tufle. Sorry for those who are disappointed now. And only those populations, because we've just started the analysis, okay? But what we can see here, and this looks very strange, but whatever, we will just continue, is that we have here the differentiation of the populations. And actually in this figure, you should see a lot more variation. I don't know why the computer is not displaying this correctly, but we will continue anyway. And here, you can see that there is basically very few genetic differentiation among most of those populations. So they are genetically very similar. There's still some differentiation, but only very little. But what you can see is they are totally different from the Mediterranean populations, from the Chioccia and Mapicolum. And the other effect that was really striking us is the Tvermine, which is exactly there in this Baltic Sea, where we have a very low salinity, right? Is extremely different from these other fully marine populations and also the other Baltic populations. We were like, wow, that's extreme, because imagine the Baltic Sea is only 10,000 years old. How can so much genetic differentiation actually evolve within 10,000 years? So we wanted to know more the details. But first, focusing on one of the other extreme environments, the Mediterranean Sea, because also there we are suffering very high temperatures and high salinity, I would say, but they are the experts over there. And what we did is that we looked at genome-wide FSTs, so how different actually the sites are from each other. Here is the genome, basically. We all see the different sites of genomes and genes that are actually very different in terms when we're comparing them to other populations. So we took Falkenstein as our basic population because this is just the place we're working at. We should do this different in the future, but just first insights, okay? So you can see that Falkenstein, St. Malo Bay, we have some differentiation. There are many genes that have a high FST, and when we then do an enrichment analysis of those, we see the genes that are actually differentiated between St. Malo and Falkenstein are involved in development, metabolism, behavior, neurogenesis, and oocyte maturation. When we then go to the other comparison, focusing on Falkenstein versus Chioccia, what we can see there is that we have more differentiation, a lot more differentiation, so we only took genes into account that had a much higher FST than here. And when we then did an enrichment analysis, so we really looked what kind of pathways are actually potentially function behind those genes, then what we have found is it's not similar pathways, but they're also very different. So it is stress responses, embryo development, and innate immune system, which might suggest that the Konschak population is under stress, and that there a diversification, a differentiation in the stress response and in the immune system was necessary to cope with these extreme conditions in Konschak. Because I think this is also a highly polluted area, which is under enormous eutrophication. But correct me, the Italian people, if I'm wrong. Is it correct? Okay. Because when I was once there, I've seen things that was not so pleasant only. Which means that potentially they have really locally adapted to this extreme environment there. This is what is indicated here. But then, the next thing that was very striking is exactly this potential adaptation to the Baltic Sea. So the Baltic Sea, 10,000 years old. I think the Mediterranean Sea is something like 3 or 4 million years old. So totally different evolutionary timescales. But when you look at this, it's massive. So there's so much genetic differentiation between the Falkenstein population that actually is also only exposed to 18 PSU, so we're not talking about a fully marine population here, and the Tvermine population, which is just enormous. How could this happen? We were really like, what did we do wrong? So we went to blast some of the genes that we have found, and this is, for example, now you can't see properly. It's RAG1, it's an immune genes, and just blasted like what species is actually matching to. And what we have found is the major hits were Syngnathids tufle, 99.14%, so it must be Syngnathids tufle, fine, all good. But then our study continued and blasted some mitochondrial genes. And what was the hit? It wasn't Syngnathids tufle, but it was Syngnathids rostellatus. So another species that also occurs there in Tvermine, in the Baltic Sea. We were like, oh, wow. Because this actually indicates, most likely, that while we have Syngnathids tufle on the hit of the nuclear genes and Syngnathids rostellatus on the matching mitochondrial genes, then most likely hybridization has occurred because mitochondrial genes can only be inherited from their mothers. So mitochondria are directly inherited maternally, whereas the nuclear genes are, of course, coming from both sides. And this is actually supporting results that we have recently collected in Falkenstein, so the place where our fish are actually living. Where we are doing our main research, that's what I want to say. Because here we had several clutches of the same male that looked like this. So we had some of the embryos that very much looked like Syngnathids tufle, whereas other embryos very much looked like Syngnathids rostellatus. And this suggests that potentially it is a strategy of those fish to hybridize to be able to cope with these extreme environmental conditions. But this still needs a lot of research, really, just the first indication. But we think it's very, very cool. And we are convinced that in this enormous data set of more than 1,300 genomes, we will have many more of these fascinating stories that might also help us to protect the species better in the future. So, taken together, I mean, those are currently on the sequencer, so in a couple of months I will be able to tell you more if this is really rostellatus hybridized with Syngnathids tufle. But taken together, we think that we can show that genetic differentiation has occurred in the colonization of the Mediterranean, and that their stress responses are really changed, as well as immune system adaptations we can find. Then we might see that we find a hybridization of Syngnathids tufle and rostellatus in the Baltic Sea. And I think what I could not address in this population genomics part yet is what's the role of phenotypic plasticity? And in my view, this is really something where we have to go to also understand short-term acclimatization better and to focus more on the role of gene expression and epigenetics in such environmental colonizations. And I totally think that you will be with me when I say we need Eurosync 2.1 reloaded that needs to focus more on phenotypic plasticity. So Nuno, I hope you're up for this. Thank you.